Good morning to you all. Respected Hari Krishnan sir, are you listening to me?
Yes, Manav, good morning.
Good morning. Okay, sir. May we start?
Yeah.
Thank you, sir. So, good morning once again, dear participants. We have among us as a speaker Dr. V.S. Hari Krishnan, who is presently working as Scientist E in the Institute of Sri Siddharth Regional Institute of Medical Science and Technology, Trivandrum, Kerala. He had his BVSc and EE from Kerala Agricultural University and completed his Masters in Laboratory Animal Science from University of Copenhagen, Denmark. He had been working with laboratory animals since 2003. He won several awards in the field of laboratory animal science, and one of the significant awards is Japanese Association for Laboratory Animal Science Young Scientist Award 2009. He has over 30 peer-reviewed national and international publications to his credit, and had presented in international arena widely, like Federation of European Laboratory Animal Science Associations of Europe. His first area are surgical animal model and animal welfare, whereas he is currently working on refinement of spinal cord injury rat model and focuses on pain and welfare. Today, he will be presenting among us on the topic experimental animal selection, care, management for various studies and regulatory aspects. So with this brief introduction, I would like to welcome Dr. Hari Krishnan sir as a resource person of the second last day of web course. And before handing over the session to Hari Krishnan sir, I would like to announce one information to all their participants that those who are interested to have a direct interaction with Hari Krishnan sir can put their name and designation in the chat box before the question answer session, or they can also put their queries in the chat box.
Thank you so much, Dr. Manav, for giving such a nice introduction to me. And I look forward to fulfill your expectations for the coming maybe 60 to 90 minutes of my presentation. So today's topic is the selection, care, and management of experimental animals. So before we start, I would like to tell you who I am. So I am a 1997 admission graduate from Kovas Manuti of Kerala, who had passed out in 2003. And in December 2003, I joined Sri Chitra Tirunal Institute as a veterinary surgeon in the coronary stent evaluation program in pigs. So this was the first time when I came to know about biomedical science field and animal models. So what I was doing during that tenure of my first stint of my professional exposure was that, as you know, heart supplies blood to the entire body and tissues and organs, whereas The heart muscles also require fresh blood and oxygen, which is and nutrients, of course, which is supplied by the coronary arteries. So there are three coronary arteries: the left anterior descending, left circumflex, and right coronary artery, which are the three major arteries in humans. And it is almost all the same in pigs as well. So we picked pigs as model to study the efficacy and safety of the coronary stents or drug-eluting coronary stent. And what happens in coronary arterial disease is that if a big segment of the coronary artery is atherosclerotic and gets its patency lost, so the part distal to that supply region will turn ischemic and the tissues can die and as a result heart attack or cardiac arrest happens. So if it is a big segment, you have to go for a what we call as cardiopulmonary bypass surgery, whereas if it is a focal lesion which surmounts up to certain parts of the artery, then using coronary stenting system, which consists of what we call as catheters and pass through the major arteries or peripheral arteries through the femur or through the carotid. It reaches here and it sees the lesion and places a biomedical device known as coronary stent. So this stent will be deflated and will be crimped upon a balloon catheter, and it reaches this lumen and then it with some pressure, if you apply some pressure in the outer exterior tip of the catheter, it inflates the catheter and delivers the coronary stent, and so the lost patency will be back. So that is the way how in humans coronary stenting is being done, and it is how coronary stents are being used. And it is just as the same as we could see in a human angioplasty. We call it as percutaneous transluminal coronary angioplasty, which is also being done in the pigs. And to evaluate the safety and efficacy, we did lots of porcine studies. So it was an Indian-made drug-eluting coronary stent, and the data they used to export it later on to Europe. So after that, I worked with rodents and rabbits since 2006 in the areas of facility management, quality control, breeding, care and welfare, and creating surgical and non-surgical animal models for various research programs. And I did my master's from the University of Copenhagen, Denmark, from 2010 to 2014, occasionally traveling to Copenhagen and attending the practicals and examinations there. And currently I'm also doing from the same university, from another department, a PhD on the refinement of spinal cord injury rat model. So this is who I am, and the agenda for today is the selection, care and management of experimental animals. So for presenting it, I put it this way, but while discussing, we will discuss about the selection of animal models finally. Before that, we will see how we can offer the care and good welfare and management to these experimental animals. So in the care and management session and selection of animal models, I have divided the presentation into two broad areas. And in the care and management, we will deal about or discuss about in brief about the areas in the specialization of laboratory animal science, and then we also will discuss about the normal behavior and the deviations. So only if you know the normal behavior of these animals, you will be able to understand what the deviations are, or you can assess the health of these animals. And then we also will discuss about how to ensure the proper care in laboratory animal facilities briefly. And in the selection of animal models, I am going to present a working examples for your thoughts and your workup later on. And we will also discuss in brief about 3 R's, a systematic approach towards animal experimentation. I expect all of these to end up at the maximum by 90 minutes. So before we start, I will have a few words on why we require animal experiments. So we require them because there are lots of unsolved mysteries, like we don't know about the cause, even the cause of syndromes like cocaine syndrome and several other diseases like several other autoimmune diseases also as well. But we also require these animals to produce vaccines of the diseases which we already know about. And as you know, you require not only a single animal study for each batch of vaccine, you require another animal study as well for proving its efficacy as well as safety, and to develop new vaccines for evolving pathogens. Like in the case of COVID, also you require to use a lot of animal studies, and that is why we are doing animal experiments, which all of you, I'm pretty sure that you all will be knowing about that. So the areas in laboratory animal science field, ultimately it is all about experimental data and welfare. So finally, we are all selfish and we look forward for benefits from each and every deed which we do. And it is not different in the case of experimental animal studies as well. We look forward for useful data. And at the same time, as veterinarians, we are also inclined or supposed to give good quality welfare and care, because we use a lot of animal suffering in between what we do to tap the experimental data from these animals. And in basic laboratory science is dealing all about with the management and care of these animals, whereas in the applied laboratory animal science, it discusses more about using animals for vaccine production, for biomedical research. For each scientist will have their own area why they are using experimental animals, which is the applied area of laboratory animal science, whereas in the basic laboratory animal science, we are looking more towards its care and welfare. Why do we do that? We actually have to define and refine the animals used so that we get valid data as point number one, and we are morally responsible for the sufferings and welfare, and we are all pledged to reduce or minimize the sufferings which these animals are going through. So that is why we have these two areas. And basic laboratory animal science, and in India in particular, I would like to stress upon one point, is that in India, the basic laboratory animal science and applied laboratory animal science many a times are done by the same team, whereas in developed countries or in the West, so the two different teams will be there. Those who are doing real animal experimentation to develop products and also to study diseases, they don't look into the basic laboratory animal science, because these two areas are really broad and no single person can deal with both these areas in their entire professional span. And defining and refining the animals used, the animal housing and environment is one major area, and then health, breeding and genetic monitoring is another important area. Management and quality control and regulatory aspects, like you have to be compliant with or maintain compliance with the CPCAC or the local regional statutory bodies, whereas regulatory bodies also will look into your animal facility, for which you should also comply to their guidelines as well. So in which animals are these studies done? Basically, the answer would be, my answer would be mice, because 60% is mice, and plus the rats, it constitutes to about 75%. If you calculate guinea pigs and rabbits, all the small animal species and some other rodents, it counts up to maybe more than 80% or 85% of the total number of animals used in the European Union, whereas it is not different in the case with the US as well. So we will focus on rodents quite a bit during this discourse. So rodents, when we talk about rodents, we know about mice, rats, guinea pigs, and hamsters commonly, and some other creatures like gerbils. And these animals are quite old inhabitants of the world, maybe millions of 60 or 66 million years ago. They have been with us, living in the world. And why we do studies in rodents? Because they are easy to handle, they are small. We tell a lot of things about rodents from the financial aspect, but from the biological point of view, they are quite similar with human beings, and we can mimic or at least do a proof of concept study using the smallest animal possible, which are the rodents. So I would like to tell a bit about their origin. So the word mouse and the scientific name Mus musculus, it is derived from the Sanskrit word mushik. And if you go through the history of the mushik or the rodents or the mice, the rats and mice, they had originated from Asia, and we believe that it is from India, and some literature says that it could be also from China or Mongolia. So these animals, they had quite a big history. So from India, when the Crusaders and the invaders came in ships, and while they carried things like spices and grains back to their domain of which was Europe, it traveled quite a lot from India or China or the Asian subcontinent to the Europe. And then after that, they crossed the North Atlantic Ocean to reach the United States. And it is from where we saw that the first laboratory rodent came into existence. So it was in 1906 from the Wistar Institute of Pennsylvania in USA. How it happened was that we saw that how the animals crossed the continents to reach US, and it reached America, where there were lots of betters or fancy citizens who would leave their bred rodents to an arena and let loose their dogs to hunt them. And once the person who owns the dog which had hunted the most of these rats, he or she won the bet. So that pet fancy breeding started to happen in somewhere around 1890s or towards the last part of 1800s, and then they suddenly developed, suddenly noticed that some albinos among their bred rats, and they selectively bred those albinos, which is the first laboratory animals to be used in 1906, which we now call as Wistar rats. So we are in America. So then what happened after that? So the Wistar rat traveled to many continents, and now many continents, and we got in Japan, Wistar Kyoto is there. In the Europe, again, another kind of animal, lots of colonies of Wistar is there, and now the commercial breeders are all situated in the America or in the Europe, and now they are sending back the animals to India in flights. The ones which travel or transverse the ocean through ship are coming back in flights, and of course with a big price. So this is their about their origin and history in brief, and what we have now is the behavioral video. So this is a 7 minutes video. These animals still has this video. I am using this video to tell you that these animals are still having their instincts intact. They are from wild animals, as we have seen just now. They still bear their wild instincts. So these domesticated animals were let loose into the wild to observe what they does, and as you can see, the black hooded rats, they are more close to their wilder counterparts, so they are more readily out in comparison to their albino counterparts, which are less wilder. So the wild animals, they are more readily starts exploring. And another one thing is known as behavior is known as neophobia. These animals are highly neophobic. They are afraid of any new things, which is an indication that they had been commensals with humans. They had been cohabiting this globe together with the humans, and they consider us as friends, but still they are suspicious over generations or over millions of years that they would be trapped by us. So anything new, they are really wary of. So you can see this wild mouse. So it is sniffing, and it is reluctant. Some cheese or something should be inside, but it is still highly suspicious about the new thing. So it is nowhere going near to the trap. So and it walks away. But whereas in the case of laboratory rats, which had been domesticated, it readily walks inside the cages. But still, with some sort of suspicion, if you see the rat behavior in laboratories, they are still neophobic, but not to the extent as their wilder counterparts due to domestication. And another natural behavior in these animals is profuse grooming, self-grooming. We call it as self-grooming, and also they does it with the other cage mates, which is known as allo-grooming. So they groom themselves and they groom their other cage mates. So this is readily seen in female rats, and in male rats they follow an aggression and a social behavior or a pecking order, owing to which the most dominant animal will be groomed by all the other animals, whereas the least ranked one in the peer group won't be groomed at all by others. So there is an order there. In the wild, they travel quite a lot. They always require a source of water. So water is very important for their life, without which they even stop eating. So they travel quite a lot of area to find water source, and they take a note of that. And in case if they lose this, they will try and travel kilometers and kilometers to find out some other source of water, without which they cannot live. And they are quite non-choosy eaters. Like they have lots of options to eat. They eat non-veg. They try out new food. Like they also eat fruits. They try stem and roots. And all they does is they try it and see whether they develop some sort of ill health. And if they develop ill health, they stop eating that. Another feature is that they also watch their group mates which had tried a new food and watch their behavior and their health, and after that they will try to eat it or try not to eat it, based upon which it was poison or not, or it is good or not. Another learning behavior which is of curious nature to the scientists is that they remember what happened after they had the last food, maybe even hours later. A sort of cognition. They are highly exhibiting the cognitive behavior. And this is a wild bird. So from non-vegetarian to vegetarian, they eat a lot. And in the wild, they are hunted by the predators. They have to run a lot and escape. And also they sniff the cat feces or predator feces, after which they run and escape and avoid the area. They also communicate using ultrasound vocalizations, based upon which one rat can, if it had come across with any unpleasant experience, they will send out signals which we cannot hear or the other species cannot hear. But at the same time, they're using pheromones as well as using ultrasound vocalizations. They warn their group mates to avoid traveling through the area. This behavior is known as rearing behavior. They are looking out whether the cat has gone or not. So they are highly adapted to the wild environment to avoid predators, because both these animals are prey species. And they live deep and they quite fight a lot, especially the male animals fight a lot, and it can lead to even death in the case of where one animal is sure that it had won or either it had lost. So it accepts the other animal to be dominant over it and tries to live with that animal. So these animals dominate an area. The male animals will scent mark using urine a certain area, just like the dogs do, and have a lot of female animals for them to mate inside that. So the mating behavior is done also following a pecking order, and these animals, they mate as per the pecking order. So I'm fast forwarding. And apart from the rats, the mice, they build complex nests using paper or water materials they had got. And these animals, they take care of their young ones when their litters are about to be born. They build nests, and throughout their entire lifespan, they are curious to use various kinds of materials to build nests, and especially to plug holes. This animal, that animal had been using a plastic sheet, whereas this animal is using some kind of leaves to build their nest. So whatever material which is available for them, they pick these materials, and it is in their behavior that they should always be able to access some materials for them to build nests. It is not only the females which plug holes and builds nests, the male animals also do just the same. So this animal is plugging this behavioral pattern which is seen. So once the nest is built, these animals will reside inside that. So it is in their behavior. So the communication is using pheromones, and you can see some fluorescence here, which is rat urine. So the rat urine fluoresces in if we light a UV lamp to it in darkness. So why it is so? It is so because some predators can see UV too. So it is being a prey species, and this being a predator species, the cat as well as the owls, they can send these animals, and the same technique they use for marking their area using urine, the same thing these animals, the predators will use to hunt them as well. So what did we learn? So we saw in the video that the rodents consider humans as commensals, but we consider them as pests and also as laboratory animals. Rodents know this fact historically, and that forms the basis of neophobia to rat traps and new food. They are extremely suspicious to new type of food if you offer them as well, and it is more pronounced in wild animals, and even in domesticated pet or laboratory rats, it is, and mice, it is there. They are natural prey species and avoid open spaces, and in the wild, rats is both prey and predator species. Both of these species are prey and predator species, and they eat lower animals. As we saw, they are omnivores. They also are eating non-vegetarian food, and also the rats, they hunt and eat mice as well. So why we are supposed to separate the species using rooms as per our CPCSA guidelines or as per any guidelines is that they get stressed if the mice are exposed to rats being predator species for them in the wild, they suspect that they can come and eat them, which makes them stressed. So that is one reason why we should keep both the species in two different rooms. But there is another argument that these animals, they are highly evolved in olfaction. They can smell even kilometers away. Something is there, they go and smell it out. So these mice, they are not dumb enough not to know that rats are in the second room, and they can smell them, and they will consider it as a historic data. Historically, rat smell was there and nothing happened to them so far. So why bother? So that line of thought is also there. Then why are we supposed to separate these animals in two rooms? That has another one major reason. Some pathogens, for example, Bordetella brachiseptica is a commensal seen in the buccal cavity of the rabbits. Whereas if you see or I could isolate it from the guinea pig colony, you will see massive mortality and morbidity in the guinea pig colonies, because Bordetella brachiseptica can ruin a guinea pig colony. So if you mix both the species for microbial reasons, microbiological reasons, or for health issues, we are not supposed to mix two species within the same room. And rats are less exploratory and more neophobic than the mice, which we saw. And there is a paradox, a dilemma where to eat or not to eat, to go out or not, because they have neophobia and exploration. Both the characters are there. They are curious. They also would like to find some food to run around and look out for nesting material. So for all these reasons, they will have to go out. At the same time, they are also wary about the predators, of the dangers of the predators coming up and turning up and eating them up. So they have a dilemma there, and they have, of course, some food preferences as well. So aggression is very rarely seen in adult rats, and in male rats it is common but not surmounting to death, whereas in mice it can often be fatal, and they can kill male mice if housed together, which never happens in the wild, because due to aggression each animal will separate outself, and it has room for them to escape from the aggressive other mice, so that they can run away and not come into the path of these mice. But in the laboratory animal cages, they doesn't have any these options, because it is very small, and if we house all the male mice together, we cannot afford not to do that, because due to managemental constraints we are all supposed to do that. But it is not not abnormal that if you see that the male mice is dead if you house together. That is especially true in case of some wild breeds, like some dark-coated strains like C57 BL-6J. So the basic behavior is the starting point for many tests in the pharma field, which we use for drug trials, especially in the case of testing of psychotics, antipsychotics, and antidepressants, mood elevators, and such things. So these animals are now caged. You have noticed that these animals spend almost all the day, throughout the day, fighting each other, maybe running away from predators, looking out for water, finding their food, making nests. So they have nothing to do inside these four walls of the cages, because these animals have ad libitum feed. All the time they have water bottles available, they have bedding, they have nesting materials given. So they have nothing to do. And this could lead to something known as vices or bad behavior. So we'll see them in depth, what it is all about. So what do the animals need in captivity? What can we do? This forms the basis of basic laboratory animal science research, and this forms the basis of care offered to these laboratory animals. So from animal housing and environmental health and breeding and genetic monitoring and management and QC, we have to think how the standards are tested, are the standards maintained, and is there any scope for improvement? So a good management plan tries to provide the closest possible means. So this is an example of a good management scheme of how to rear rabbits. So they are being provided with social housing. These animals are highly social. So if you singly house the animals, at least the females, they will be stressed. This is especially true in the case of guinea pigs, both in the case of male and female guinea pigs, as well as in the case of male mice and in the case of mice and rats. And in the European Union, they got if we are planning to isolate or singly house an animal in a single cage, we are supposed to justify why we are doing that. As long as you cannot do that, you cannot supposed to singly house the animals. These female rabbits, they are living in groups, and of course they got some sort of hiding areas, enrichment material, and a variety of foodstuff is being, fiber-rich diet is being offered, because if you increase the carbohydrate content of the rabbit feed, it can lead to the loss of normal microflora in their gut, and they themselves will die due to so-called enterotoxemia and some other diseases develop in the animals, and the metabolic deaths can happen. So in the case of mice, you can see a mice being housed with some enrichment material, like some coir ropes and all, and ample space for each animal to go and explore and lead their lives in an interesting way. So they can eat, and we can also provide them with hiding areas and tunnels to simulate the burrows which these animals are used to in the wild to take care of their behavior in the wild, behavioral requirements in the wild, at the same time not interrupting our managemental activities of practices like cleaning and all. So it should strike a balance where you are supposed to strike a balance between the management aspects as well as taking care of their behavioral requirements. So such a balance is where we form the guidelines and the principles around the care and management of these laboratory animals. So in this video, you can see some animals. Some animals are moving around. So this is a cage with normal animals. So normal animals usually will be very highly active. They move around, whereas this cage also has animals. They are not showing up. So they are not well. So this is the first inference which we can make from seeing these two videos, these two cages. So this is the first case where the animals are quite active. They use the tissue paper. So tissue papers can be supplied for these animals to as an enrichment material for them to spend some time. So whereas this animal, it is not moving, and it has got some slight disability in movements. And regarding this animal, it is not moving at all. It is just hiding. We don't know why, if it is due to the camera being present or not. At the same time, this animal, it is having a hunched back, not very smooth and pliable hair coat. So all a ruffled hair coat. You can see the ruffled hair coat in this case also. Some red tears are there. Some barbering has happened. Has lost some hair coat there, and it is not really looking healthy. This is a normal guinea pig, and this rat is, after the surgery, it is normal, and it had undergone some cranial surgery, whereas this animal is showing some sort of epileptic seizures. So all the conditions which we see in humans, almost all the conditions you can also see in these animals. There are some differences which we will deal through the next few slides. So this, what is this? This animal, the next video, this animal is moving around in circles. So meaningless activity, meaningless repeated activity is known as stereotypic behavior. It is seen in all captive species. If you go to the zoos, you can see some tigers pacing. It is known as pacing, walking around and around in circles as if they are. That all happens. Why? Because they lack the behavioral complexity they would like to have in the wild, inside their respective cages. So you can see the stereotypic behavior here. It is known as circling, and this is known as looping. So somersaults is known as looping. They are all behavioral abnormalities. And you can see more behavioral abnormalities in the form of bar biting. So this animal is not eating its food, it is just chewing the bars. So it is known as bar biting. It is another behavioral abnormality. It all points towards the lack of adequate welfare. So this is stargazing or try to escape behavior or stargazing. Some argue that it is the escape behavior, but some argue that it is due to reduced welfare. So this is a group of normal animals. They huddle together, cuddle together, and their skin coat will be smooth and pliable. Their eyes will be a bit exophthalmic and smooth without any red tears. These animals are really active. So you can see how healthy these animals are. They are all really active. So they try to eat food, they climb up, and these are a group of healthy rats. The exploratory behavior, and this is a group, this is a healthy rabbit. It looks really healthy, and a healthy guinea pig. So this is how examinations are being done. So you can see some ruffled hair here. So for more closer examinations without even handling the animal, you can tube it. It is known as tubing technique of handling. You can use plastic tubes and to see the animals all around. So this is another vice which we see as a behavioral abnormality inside the animal, which is known as barbering. And there are lots of other behavioral abnormalities also, but there are more. So to know that, we should know about the cardinal signs of health. Basically, we term it, I missed one important point which I will tell. So it is the clean coat, yellowish a bit in males, and in older animals are also normal. So you have to see the history of the animals, how old they are, whether they are males or females before you jump into a conclusion. So in general, it is clean coats that is a sign of health. Well-groomed appearance. So they groom like cats and other feline species. You have seen that the rats are grooming a lot. Grooming a lot, and well-groomed appearance is a sign of health, and activity and social behavior is another sign of health, and formed hard pellets and yellow urine. It is red in the case of rabbits is also okay. Normal growth curve you should see, and also you should look out for red tears. If you see red tears, it shows that the absence of grooming is there. To have red tear is normal in these animals, but to see them sticking around over their skin or inner paws, it is not normal. You can use some scales like a rat body condition score, and you can term the rats as obese or emaciated or well conditioned. For similarly, you got body condition scores for mice and even for rabbits. So rodents are notorious, as you know, historically they had been contracting us with diseases like plague and hand-tan and several other bacterial and viral and protozoal diseases as well. And we should be really running a health monitoring scheme or protocol and to avoid and detect these. Why? Because they don't exhibit clinical infections. All these infections in these animals are all subclinical. So there won't be any signs. They just look healthy. At the same time, they would be lodging some diseases. Reproducibility in research is affected by a lot of factors as well. So this reproducibility, if you look forward to a publication and you try to do your research with the same and to see similar results, and you are failing to see some similar results, is not very uncommon in the field of biological science. This happens due to underreporting of materials and methods, and also due to variables in housing conditions, as well as in the health status. So the quality is also affected by a number of factors. So as you see, all the variables which we provide to these animals are a variable. Like environmental factors, chemical factors are there, microbiological and physical factors, right from bedding and feed, which should be all under quality control. And also regarding the genetic factor, which is an intrinsic factor. You cannot do much about that. Other than you go out and buy the animals from a reputed vendor. And avoiding microbial burden, as we saw, is quite important. It is very hard to detect also. You require PCR, either PCR or serological test. Both are, PCR is not much costly, but at the same time, it is not widely available over India. Whereas serological tests and kits are there. It is highly costly, and you need to spend a lot of money, and it is not easy to go on and detect all these microbial agents without these two areas. Whereas avoiding the microbiological burden we can do using certain precautions. So there is a conventional technique of housing animals where what is conventional housing? It is just a shed or something like that where you don't have any microbiological separation from the exterior world environment with respect to the interior environment, which is not a primary and secondary enclosures. The primary enclosure is that where the animal lives. It is the cage, whereas the secondary enclosure is the room of where the cage is situated. So these the microbiological status you see in the primary enclosure won't be that much different from the environment which is exterior outside the building. So that is what conventional building is all about. It has got lots of flaws. You cannot predict what your health status is for today in comparison to yesterday. So your animal studies are not very standardized. If you house them in conventional facilities globally, we go for barrier bred facilities, but you can integrate some sort of barriers using mosquito nets and food dips and things like that, and showers and dress change and autoclave bedding and feed. If you have a conventional building, which will fall somewhere between the conventional facility and a barrier bred facility. So this is an example, a previous example of how the animals houses were barrier bred. You can see an isolator. There will be a chamber. You cannot. There are two doors, one here and one there, and inside there will be animals. You can handle them only using gloves. You can handle them, and you can supply them to the next cage where males are for breeding and all. So the breeding nucleus is entirely precious. Is being housed in such containers known as Trexler's isolators. You can open the front lid, place the feed inside the chamber, and then spray. Using your gloves and everything will be there. You can spray it and disinfect the area and then close it here. And from the gloves here, you can go in and open the next door and pull the feed to the inside of the chamber and supply them to these animals. So it is an entirely rigorous process. But it provides a good barrier. So operational ease is more with the conventional facility, and whereas barrier bred it requires a lot of infrastructure and also a lot of operational difficulties. So this is a modern day isolator, we call it as conventionally individually ventilated cages. So they also give a very good barrier. This is level blocks, so that the entire building used to be a barrier. So that had operational impracticalities, like you cannot use the building for any other things. There is less flexibility. If you want more rooms, you cannot do anything. So after that era of building level barrier, they took it down to the rack level barrier where you can find Trexler's isolators, and now it had further shrunk down to the cage level barrier. Further, you can open these are known as individually ventilated cages. HEPA filtered air will be driven in, and you can set in the console the number of air changes required per minute, per hour. And it will be extremely dry, and it reduces the operational thing, like you need only to change the bedding once in a week in comparison to every other day in the conventional facility, because it is also sucking out the noxious air. So the ammonia concentration inside the cages will be extremely low, and the microbial entry also will be low. It has got another advantage in that if one cage has infected animals, it won't cross-contaminate, because the air driven to each cage is separate and clean. And before exhausting it out to the building, they also filters it and decontaminates it so that the environment is not undergoing any harm. So there are standards based on this, and all are free to download when in a 1.5 hour lecture. It is I'm not having enough time for explaining each and every standard. So there is a guide. This is an American standard. You can go on and download a free PDF from the web. And this is the European Union guideline. It is latest revised. It is commonly known as New European Union Directive, and it is the legislation in the European Union now. And whereas in India we have a CPCSA compendium or guideline. So we as basic laboratory animal scientists, we are using lots of tests to see whether what the animal really requires to provide them with adequate care or not. So usually the temperature recommended in all these 3 guidelines or even more number of guidelines is around 22 degrees Celsius for rats and mice, whereas the thermoneutral zone is much higher in the case of rats and mice. So some scientists, these slides are to explain how we work generally. So this setup was made where 3 different temperatures was offered to 3 units of laboratory animals and a single unit of laboratory animals, and they were free to move from cage to cage using a channel. Through the tube, they can access both the areas. So what they preferred was studied. So finally, what we found out was that they preferred something like 30 degrees Celsius when they were inactive. They were inactive. During the activity, there was no significant difference. So their requirements, if we just go with the guidelines, their requirements won't be matching to the guidelines, and there is more and more research, and day by day we are having peer-reviewed data coming out to show that this field is highly evolved and dynamic. So in India, basically the committee for the purpose of control and supervision of experiments on animals, commonly known as CPCSA, gives guidelines, and this is a statutory body to which you are supposed to register if you are a biomedical research unit or education or a breeder who provides animals for biomedical research in India. And regarding the applied laboratory animal science, there are lots of areas. I'm just terming here quite a few: drug and vaccine research, medical implants, stents and valves. In education, you require animals for genetics studies, medical products like blood bag and equipment, space and defense research. You can imagine, recollect that Laika was the name of the dog which was sent by the Russians to the space. It was also a laboratory animal, actually. So in basic research, human behavior and neurology, lots of drug research, toxicology, and many more. And welfare assessment in all the experiments and presence of veterinarians during experiments is really important. So now we move forward to the next session, which is the animal models. And here the agenda is to see and discuss which animal for which study or which test, to familiarize with the terminologies including animal models and the basic concepts on selection and methods of decision making, and to generate confidence in making to take decision on which animal model will be most suitable for your animal experimentation. So if it was a real-time lecture, I would have given you with some workouts, but I will just try to illustrate how generally we are doing this in practicality. So the concept and definition of animal models. A model is an object of imitation. So if you plan to imitate anything, it is a model. And if you plan to imitate a human or an animal condition, it is an animal model. A lab animal imitates a biological phenomenon that the species has in common with the target species. Modeling of humans and also, of course, other animals for vaccines. For example, Mendel studied peas using fruit fly, which was his laboratory animal for learning human genetics. So we can try to see what are all the classification of animal models. So there is a class known as exploratory models. So you don't know about a mechanism. What happens if you if the COVID-19 attacks the body, what happens? You don't know. So you require an animal model. It is a novel virus and you still don't know what happens. So it requires an animal model which can simulate the system of a human so that you get to know how exactly it works inside the body, what deleterious effect it exerts inside the body, so that you can tackle it in a better way in the human. So it is known as exploratory models, explanatory models. So now you know something about what COVID-19 does to the human body, but you have to prove that inside animals. So for that you require an animal model again. You have some hypothesis inside you. You have to prove that. You need an explanation why it happens, for which you can use an explanatory model. And a predictive model, extent of resemblance of biological structure of animal with the corresponding structure of humans, is termed as fidelity. So this is another important term. So high fidelity models means close resemblance. So there are two more terms known as when we talk about fidelity, there are two more terms known as face validity as well as construct validity. So these two terms are quite important in terms of fidelity. What are they? Face validity. I will tell an example. Please don't read through all these things. I will just explain. The face validity, if you have some experience with laboratory animals, you know about a disease condition known as mousepox. So mouse different pox lesions. Humans also have chicken pox. So if you consider using mousepox as a model for chicken pox, so we can call it as they both are seemingly just the same. So they both are having high face validity. At the same time, they are having very low construct validity. Why? Because mousepox and human pox have different pathological reasons, because they have two different etiological agents causing disease in both these animals, in mice as well as in humans, which makes them entirely different disease. So seemingly same, but not really. So that is the difference between face validity and construct validity. Another one example is WHHL rabbits. Watanabe, a Japanese rabbit-bred mutant rabbit strain is there known as WHHL rabbits. Watanabe, heritable hyperlipidemic rabbits. They develop hyperlipidemia, ischemia, and atherosclerotic plaques inside their vasculature or vessels. Are they really close to humans? It is not. Why? Because in the nature, rabbits are herbivores. At the same time, WHHL rabbits had been given with high fat diet, and they are no more herbivores. And the way by which these animals pick up atherosclerotic lesions are not exactly the way how the humans pick up atherosclerotic lesions. So construct validity is very poor. Whereas you see HACEK knockout mice, HACE2 knockout mice for COVID-19. So this three, by mistake I put the three HACE2. I have corrected it now. So HACE gene 2 knockouts are being widely used for COVID-19 research, because ordinary mice with the ordinary ACE2 gene of mice, COVID won't attach to that region. Whereas that region is spliced off and integrated with a human ACE gene to create a knockout. It is known as a knockout technique, and it is known as knockout mice for using which COVID-19 models are being tested. And it is having high construct validity with the humans. So isomorphic animals models have face validity alone, whereas these are basically the basic classification of and terms involved in animal models. And regarding disease models, there are induced or experimental disease models using surgical, chemical, pharmacological, or nutritional means. Whereas there are spontaneous or disease models. Basically, these animals are all created, human-made. And genetically modified models are also models for spontaneous disease. It comes as a subclassification, but on its own itself is another class. And negative disease models, certain animals does not, even whatever you do, they does not produce a disease condition to simulate human condition or to simulate other species condition. So such animal models are known as negative disease models. And there are orphan models. For example, some diseases don't manifest at all in humans. So animal models are used for the benefit of other birds in the case of Marek's disease, where vaccine production is aimed for. So you can read more from this regarding the classification of animal models. And now we have got some idea about, I am completing 1 hour now. Maybe in the next 20-30 minutes, we should be able to wind it up, including the question and answer session. So extrapolation, phylogenetic relationship is really important. It should be phylogenetically related to human beings. At the same time, if you argue that it is for extrapolation, you require all monkey studies because they closely simulate humans. It is not really ethical or legally approved to use directly plan your animal studies in monkeys, whereas you should do it in the least sentient or the sentient species, which are the mice, zebrafish, mice, or rats, and higher. So the phylogenetic relationship is there, shall be considered. For example, thalidomide crippled. This is a classical example. 10,000 children or even more are not causing toxic birth effect to rats. The thalidomide was tested in rats, but at the same time, it ended up in a tragedy. So it tells us that even if you do studies using animals, you cannot be 100% sure about the safety, for which after this incident, the scientists, they are using more and more stringent terms, multiple species, non-rodent species studies, rodent species and non-rodent species, a higher animal model, and limited clinical phase trials, and then to human beings. So that is why the COVID vaccine is taking much long. So anatomic and phylogenetic physiological similarity, but biochemically different. So some animals will be in anatomically and phylogenetically and physiologically similar, but biochemically different. So biochemical similarities, there is a table. If you see that a textbook, there is a table I can send you if you ask me later on. It shows that the species wise, how many, which species is closest in enzyme systems wise with humans. So if you see that dogs are closer, monkeys are really closer, rats are not bad, rats and mice are not even bad. So for some systems you can just use and conclude it with rats and rabbits, provided if you can prove that they are close enough to the human or the target species. So to which humans? So if we are telling about Indian race or Africans or Chinese, they are all different. Chinese are prone to some things like sickle cell anemia or to some other diseases like ethmoid tumors and all, whereas some populations are not exposed to or vulnerable to those. So population-wise study is also required when we go from the animal trials to the humans. Here is another classical example. We have learned that pigs are a good model for blood vasculature and cardiovascular system. If you consider an artificial heart valve, a prosthetic heart valve for pediatric purpose, and if you try to use, because we already have the data that the pigs are quite close to humans, and if you are selecting a piglet of maybe 4 kg, 5 kg size and implanting it with a heart valve, do you think that it is a good model? Actually, even if all the parameters like blood vascular system and all the micro enzymatic things are all similar in the case of humans and pigs, you cannot do this study. Why? Because the growth chart is, if you look into the growth chart, the pigs gains maybe 100 kilos in 6 months, whereas the human baby will be by 6 months 2 kg. So the heart size also will be bigger, and there are chances that the experiment can fail owing to the sutures being torn off due to the exponential increase in body weight in the pigs. So you should consider each and every point before jumping into a conclusion. So the various animal models use are transgenic, normal. Transgenic, you can also have another thing known as knockouts or knock-ins. Transgenic integrates a gene from another species, like human gene, like as we saw in the HACE2 mice, whereas knockouts and knock-ins, they either knock out a gene of interest or integrate a gene of interest from the same species to another position in knockouts and knock-ins. And normal or naive animals, we use naive animal studies in regulatory testing like toxicology studies and in lots of preclinical trials. Normal animals itself are sufficient for you to publish. That the normal animals are basically used by the regulators or regulatory bodies recommended studies or tests. Drug-induced animal models are there. Surgical animal models are there. You can ligate off the entire femoral artery and create an ischemic model. So you should all see about what is the difference between the blood vessel, that regional supply that is there in the animal model plant in comparison to the humans. And in some animals, if you tie off or ligate off the vessels, collaterals develop. Prolific collateral vessel formation is a characteristic feature in animal models in comparison or in difference with in contrast with the humans. So you cannot use that animal models to predict, but for proof of concept studies or things like that, you can categorically use these animal models. And mutant models are there. One such example is spontaneously hypertensive rats. I have told about how the Wistar rats traveled all over the globe. Wistar rats also reached Japan, and in a lab known as in a lab in Kyoto, they developed a model, a strain known as Wistar Kyoto. It is quite different from the original Wistar, maybe by line breeding and segregation from the original gene pool. And what happened in these animals is that in spontaneously hypertensive rats, they've found out that all of a sudden some animals showed accidentally, while they were monitoring for their particular study, they noted that some of the animals were having high blood pressure, and they isolated these animals using a truncation selection process. Beyond this, they set up 150 mm of Hg as a baseline, and all animals having a blood pressure over that, they isolated and selectively bred. And because of the mutation, it happened, and this model is still widely in use. It is a mutant model. It's an example of a mutant model. And in nutritional research, by depriving them with vitamins and minerals and things like that. In nutritional research, for example, if you plan to use rabbits as a B-complex vitamin deficient model, you are likely to fail because they produce vitamin B-complex and they ingest their own fecal matter for them to re-ingest and re-ingest micronutrients and other nutrients and B-complex. So there are lots of and especially if you deprive the guinea pigs with vitamin C, from vitamin C you are supposed to see scurvy, as you can see in humans, as a result of reduced vitamin C concentration. So minimizing to find the effects or results, good results from animal models, the basic principle is to reduce the variation. If for you to see the same effect with same size, if the variation is bigger, you cannot see the effect at all. So there won't be any statistical significance. And to reduce variation, people use inbred animal models, whereas outbred animal models are used to simulate human studies, because human population is highly heterogeneous. Whereas lowering the number of by using inbred models, you can also lower the number of animal models, or the sample size can also be reduced. Because of low variation, you are likely to strike the same statistical significance with lesser number of samples size. So lesser number of animals can be used to prove your point. So this is the last part of the presentation. So this is to consider yourself as yourself as a in the seat of a biomedical researcher or a scientist planning your own study, or you're handling someone other's study with a requirement that an animal model has to be, someone approached you with a question that an animal model has to be developed to study intervertebral disc degeneration and its therapy. So for this study, how will you plan a study? So this scientist had got one more condition, telling that the research group requires 0.5 ml of blood every day for the assay. So how this issue or task can be approached? So you can once again see an animal model has to be developed to study the intervertebral disc degeneration and its therapy. So what has to be done? So point by point we will discuss. So first of all, you have to see whether an animal model exists already for this or not. So using a literature search, an intervertebral disc degeneration study and its therapy. So you should go to the second part, the therapy post, or the therapy part, where the scientists claim that it is a novel therapy. You should also Google that and see that it is novel, genuinity is there, without which you are wasting the number of animals and your time, because it is already a proven point. So to start with, for both these aspects, for you to search the failure, it is also important that you search literature to know about failed models. So it is why it is important in biomedical research for you to publish even negative results, because your failure saves so many lives of laboratory animals, so many sufferings of laboratory animals, as well as so much efforts and cost of other scientists as well. So using a literature search, you can see whether the study has some novel objectives and its therapy is novel. If not, reject the study. And the 3 R's is to avoid experiments. Also required for you to understand 3 R's. What are the 3 R's? The first R is replace your experiment. So as part of replacing it, you are doing a literature search for a start. Why? Because you can itself at that stage itself find out that either the drug or the therapy offered already exists in market, and you can also question if the same scientist tells you that still they require to do that because it is an indigenous product. You can make that point valid in your application. So first one is replace your animal studies using a literature search, and then with some cell culture studies you can drop this material into a cell sheet to see whether the intervertebral disc cells are getting some good results, or whether it is cytotoxic or not. So some sort of in vitro studies forms the next. You cannot term in vitro studies as a total replacement, but at the same time it is a major replacement. Why? Because for cell sheet studies also you require cells, and from where the cells are taken, again from animals, but you can use a very few number of animals to do a lots of cell sheet studies. And then comes reduction of the animals. If you had decided that from the literature search that it is a valid study, how to complete your study with the least number of animals at the same time getting statistically significant results. For that you require to focus upon reduction. And then if you have chosen the right sample size, how to inflict minimum damage to the animal, minimum pain and welfare issues to the animals, at the same time get valid, statistically valid data and good quality data. So that is refinement of your experiment. So is it a regulatory study? You should ask, and if it is yes, you can. If scientists or we want it to do as per OECD or ISO guidelines, we don't have much options left with us to choose the animal model. Why? Because in the standards itself, they will tell which animal model can be used, whether it is females or males can be used, what shall be the sample size per group, and how the study has to be done. Everything will be in the guidelines. So it is made very simple. Whereas in the comprehensive and in the literature search, you are supposed to do just a Google search, a simple Google search. You are likely to miss much of the significant results. Whereas a search engines like PubMed search using medical subject heading terms or mesh terms to populate all the possible models and their pluses and minuses. So you get a population of with some training. If you do a good PubMed advanced search, you will populate the entire studies, and then you can screen each study and you can find out which animal model is likely to be successful and which animal models with good results had been published, which are the animal models with negative results or negative animal models, if some are there. You will come to know about that after a comprehensive literature search. And then you should evaluate the therapy. How much you should ask, you should go back and ask the scientist how the therapy is done. If it is an injectable, how many times you have to inject. If it is an in situ cell dropping or something like that inside the intervertebral disc, it is a surgical process. So you should know about all these things. And if it is a surgical model, how much volume is required of that material, particular therapy to be injected into the site, so that if it is much, you might require at least rabbits. If it is very, very, very small, maybe very few microliters, you can end the study up with rats, even with rats. Number of injections required to treat the IVD. So these questions you shall ask, and if you find everything, you have a condition put forward by the scientist. Requires 0.5 milliliters of blood every day for their assay, maybe for 30 days, 40 days, you should ask. And even if it is for 2 months, you have with syrup, even if it is for 1 day, you cannot use rats or mice, because 0.5 ml blood withdrawal and making the animal survive is impossible in the case of mice and in rats. So you can conclude that a rabbit will be the minimum possible model or the lowest sentient species model suitable for the study. And further, again, you should go and evaluate the rabbit model, read more on rabbit model and the materials and methods section and regarding the construct validity, the cell structure or the intervertebral makeup of embryology. From embryology, you should read of the IVD disc makeup of in rabbits, whether it can simulate rats, humans or not. And after asking all such questions, you can finalize your model. If you still find that rabbit is not a good model, you should again do the entire exercise once again until you find the right model. So some skill is required in experiments and welfare. So I am also incorporating some slides here. So pain assessment is in rodents, stress assessments in rodents, you should know how to do that. Negotiation of pain and minimizing stress, experimental models. So how can you acquire these skills if you are new? Education and or training. So from where to for you to get further education? So there are FELASA or Federation of European Laboratory Animal Science Associations categories known as functions, and these it is available. So this is one such training available from the University of Copenhagen. It is all online, and I am an educational coordinator. If you wish to join, it creates certified designated veterinarians who can do animal studies and maybe for your higher education. So you can find some scholarships as well from, for example, ICLAS scholarship for Denmark. This particular course is being funded by E-Class Scholarship for veterinarians in laboratory animal science and medicine. You can try and apply. Try even someone from Pakistan had been awarded. So it is our chance now. Maybe some of you can go on and apply the E-Class Scholarship. And also the Laboratory Animal Science Limited, Laboratory Animals Limited, is a registered charity working from United Kingdom, and they will give you individual funding to attend training courses. So you go here and click here, and you will find this, and you can try your luck there. So if a FELASA undertaken, or it is a FELASA certified program, so you should be able to find some scholarships if you are lucky enough to from Laboratory Animals Limited as well to attend the course. You can be at your home and do the entire course and pass an examination. So UFA grants are there. So there are some Indian trainings also. They are certificate course, 10-day certificate courses available from Tanuvas or Tamil Nadu Veterinary College, Madras Veterinary College, and training in Kerala. We are offering twice in a year, extremely cheap, maybe 3,500 rupees for 6 days. We provide you with working lunches and tea and maybe an accommodation for 250 rupees per day. And it is a basic, very, very basic level of training. It is not advanced level of training. It aims to educate and train candidates who are just beginners in the field and non-vets, maybe non-veterinarians. Maybe there are some veterinarians who can join are also welcome. But don't expect that it is a high-end training, because it is usually held for MScs and PhDs. But maybe in the future we run specialist training programs for you as well. So you can also, being in your home, you can train for free. Go to the NC3R's website, and starting from how to pick up a mouse and to assess the health and ill health, everything, they are providing free modules. You can get yourself video trained. You cannot lay your hands on a rat or a mouse, or you can just get yourself satisfied with your computer mouse. So this is our facility. It is again having individually ventilated caging system and a controlled environment. So this is I'm done with my presentation. It is over to Mana, and for you to ask some questions if you wish to do that.
Thank you, sir. It was quite interesting and informative too. And probably the participants has also enjoyed it. And there are quite a few numbers of questions as well as appreciations. You can also check it over the chat box, sir. So if you allow me, we can go for the question and answer one by one.
Yeah. So can you ask which is the first question?
Yes, sir. The first question is from Anjan Jyotinath. You have to scroll up.
Yeah. Anjan Jyotinath is my friend, colleague, senior colleague, my friend colleague. Yeah. Nice
Yes,
to see you.
He is presently working in our college in the Department of Microbiology.
Nice.
So,
I wish I could visit you once.
Yeah. Dr. Anjan Jyotinath, if you are there in the Google Meet, please unmute yourself and you can go for the question. Probably he is not there. So
No problem. Can you read it? Read his question, or I can answer him later.
Okay, sir. Should I read it?
Yeah, please. Which one?
So he wishes good morning to you. Then he praised your presentation, and he is asking, what is your opinion on genetic monitoring of experimental animal in Indian perspectives? Then the management.
It is a very good question. It is a very good question. Actually, to answer that, he should also have been here, because we can together work out wonders if we work in coordination. In India, basically what we lack in general is genetic monitoring as well as health monitoring, and we cannot suppose each and every facility to have these facilities. So we shall all be together to plan a facility for genetic monitoring and health monitoring as a centralized facility so that we can get our animals tested. Actually, even if we breed all for ourselves, my historically bred Vistar won't be your Vistar, and it will be both of our Vistar won't be the same as the original Vistar which is available in the US or in the Europe. So what they do is a periodic or planned circulation of breeders to maintain their gene pool stable. So as long as we are not doing that, we cannot claim that our Vistar is a Vistar or a Sprague Dolly is a Sprague Dolly. So if in order to do that, we require genetic monitoring, and it is quite expensive for you and tedious to us to integrate that. And to replace it, as a suggestion, we can all team up and set up a centralized facility or apply for a funding. It is possible.
Thank you, sir. And I'm shifting to the next question from Dr. Muhammad Iqbal Yatu. He praised your presentation, then he wrote, a large animal experimentation, though imperative many a times, but regulations are creating hindrance. How to overcome such issues?
So now, Dr. Iqbal, now CPCSA had undergone vast changes or sea changes, I would say. They are approving large animal protocols within a month. Or currently it is 2 months since the past 1 year or 2 years, and they are claiming that, or they are promising that they have again revised their speediness or their working time and had reduced it to 1 month. And if you submit it and if you pay 5,000 rupees per animal large animal form, they are promising that they can sanction it in a month's time. So things are being changed. Things are getting changed, and they are coming up with very good things now. That's what I could see as an IAC member secretary as well. So we are doing a lot of large animal studies. We got approvals for each and every study. They had not rejected even a single study. Maybe some number of animals gets reduced, but that all depends upon how you can justify your sample size in case of large animals as well. Hope I cleared your query.
Yeah, I hope so, sir. And there is one more question from one of the Central University of Assam. He wrote, sir, while experiments being carried out with mice model or any other animals, is there any changes in their behavior while being caged, rather when they are set free? And if it affects the experiments, if they changes the behavior if kept for a long time in cages?
Exactly. So the thing is that if we have seen from the rat behavior video or the mice behavior video that they are getting lots of freedom and free space and time for them to do lots of activities, and when they are caged, they are being deprived of all these freedom as well as avenues to carry out their species. Even their species specific behavior gets hampered. Expression of their species specific behavior gets hampered. So how can we overcome that? That's what we were discussing through the first half of the presentation. So there are lots of ways, enrichment items. So anything doesn't form an enrichment. You should give them only with validated enrichment. So if you are providing them with something like a piece of paper or tissue paper, autoclave, you can autoclave that. It has it had been proven that it could be really beneficial for them. So I have some cardboard tubes and things like that. Even coir ropes tied from the top grill to bottom. They will climb up, they will shred it. So what do they do during all the daytime is just do nothing. So we are creating validated avenues to simulate the wild. At the same time, you cannot place mud and things for them to burrow because it will hamper the cleaning and management activities. So there should be some balance. And that is why validated enrichment material, at the same time, it should also be cheap and available in India from an Indian perspective. So if you can provide them with that and make your animals more happier, I think our data will also be get benefited from that.
Thank you, sir. Shifting to the next question.
Okay.
He also praised your presentation from Mr. Ranjan. Then his query is, what are the regulatory system for lab animals care, management, and experimentation in India? Probably you have mentioned in your presentation.
Pardon. I didn't get to Mano. Can you repeat, please?
What are the regulatory system for lab animals care, management, and experimentation in India?
Okay. Yeah. So we got a single agency handling all these things, all these issues. It is not a CPCSEA. So if you remember one of my slides, I showed the website of CPCSEA, and if you go to that website, you will find a thing known as a PDF known as, of which I showed the photo, a PDF known as Compendium of CPCSEA. It is free to be downloaded. And if you can download it, and if you press the Control F button, and if you find Compendium, you will find, or guidelines. If you type guidelines, you get a set of guidelines, and those guidelines are only the guidelines which we are bound to be adhering to, and that they controls and supervises and regulates all the experiments in India. I think I answered to your point.
Yes, sir, exactly.
If it is something else, don't feel bad to write to me as well.
Okay, sir. So he will. And the next question is from Dr. Snigdha Hazarika. The question is depression level in mice is comparable to human beings? What is the external behavior of a depressed mice?
So depression and fear and anxiety and exploration, these behaviors, and of course cognition or learning, these behaviors had been widely studied using rats and mice, and many of the drugs which we use as antidepressants and things, mood elevators and antipsychotics, these had all completed their trials and evaluation using rodent species, especially mice and rats. So we can very well take that they can give valuable conclusions or inferences on the behavior. At least they can tell us that they express fear for predators. So there are lots of behavioral tests devised to test whether the rodents are fearful or the rodents are fearless or more exploratory in their terms. And using these tests, they could generate data, and this data is used to prove many of the drugs which humans are using. Of course, after this process, these drugs again will have to go through a limited number of human trials, and then once it again gets valid results, it finds the market. That's how it happens.
Thank you, sir. The next question is, the participant wrote, sometime I have observed that Swiss albino, both male and female mice, shows cannibalistic behavior in spite of providing them with standard diet. housing and environment. What could be the reason for such cannibalistic behavior in caged mice?
In caged mice, actually, it could be due to several reasons. Maybe if you got noises, it is proven that the first luxury these animals will shed off if they are coming across with any sort of stressor is their breeding. So they turn cannibalistic as well if they are stressed during or agitated or too many changes. Too many cage changes could be a factor. Using corn cob, you can reduce the number of cage changes, and we have also seen that in certain mice for the first 1 or 2 breedings, and it also holds true for in the case of rabbits as well. So in rabbits and these mice, for 1 or 2 breedings, the first 1 or 2 litters, cannibalistic behavior can be seen, and it is more often seen in mice and Balbsi inbred animals in comparison to the outbred animals.
Thank you, sir. And the next question is from one of our regular active participants, Dr. Zayar Khan, sir. His question is, what is the normal body temperature of rat, mice, and guinea pig? Whether thermoneutral zone is same or different?
Thermoneutral zone falls something like 30 to 34 degrees Celsius in these animals, especially in rats and mice, and the body temperature is comparable to that of a human being. And in rabbits and guinea pigs also, there is not much difference because they are all social animals. And in the case of rabbits, you cannot house them in groups because the male rabbits will fight, and the female rabbits can be grouped in groups, group housed in groups. And it differs in the case of whether if the animals are in groups and if they are individually in a single cage. Why? Because the animals huddle together and conserves heat if they are group housed. And again, it also differs if the mice is nude mice or if it is having fur. So during the active phase, it is seen that they prefer 30 degrees Celsius and 22 degrees Celsius. Why it is given is because in the West and in Canada and in the developed world, there are regions where animal facilities are located in minus 20 degrees Celsius during the winters, and they have to heat it at least up to 22 is a good amount of energy usage as well as a satisfactory amount of heating up for them. Whereas the temperate countries like ours, we need to cool it down to 22, which is again up to 22 is just okay. So it is to my feeling, it is that they had stricken a balance between both the regions and while they were deriving the standards. A much more temperate climate they will very well tolerate. Why? Because 30 degree is the average thermoneutral zone, or even a few more degrees more in the case of rats and mice. So they can tolerate it, and they love temperate atmosphere because, as we saw, they all originated from India or from this subcontinent and travel. They might, and of course they can tolerate a wide range of temperature. But of course, there are papers showing that 22 degrees Celsius can add thermal stress or cold stress to them. There are papers.
Thank you, sir. Yes, sir. There are quite a few numbers of questions. If you are okay, we can continue.
Okay, we can do that. If there are quite, how many questions do we have?
I think 2, 3, 4, around 6, 7, like that.
So can I answer them one by one and send it back as a mail? An email would be nice because I had been talking for the past 1.5 hours. And if it is
Sure,
6,
sir, that can be done.
I can definitely contact them, and I have also given with my email address, and if possible, you can they can feel free to write to me regarding their queries, and if I can answer to my best possible. I will do that.
Yeah, I think that will be better, sir. So we will send all these questions to your mail, and you can present them or answer them accordingly.
Thank you.
And then I think we can conclude. And I'd like to thank you personally, sir. It was very informative and nice lecture. Even I have learned many new things, I should say, from your presentation.
Thank you so much, Dr. Mana from Assam, to contact me and let me do this lecturing to you. I'm glad to hear that you got some benefits out of my lecture. Thank you so much.
Definitely, sir.
Hello, can I have one question last?
Yeah, please.
For repeatability of the results, how much minimum animal we should use? I'm Dr. Khan. For repeatability of
Regulatory
research purpose, I'm interested in growth hormone in animals, so I want to use the rats. So how many minimum rats should I use?
So the voice is breaking. I'm not sure.
For reproducibility of the results, can you hear me?
Sir, may I interrupt? He wants to ask for better reproducibility of a hormone-related experiment, how many numbers of animal the participants need to take?
Correct.
So for a hormone study, right? So it all depends upon what hormone you are studying and what is your animal model first of all. And if it is a very specific mutant model using which you are doing the study, you can there are many variations. So here you got, it is as simple as 6 variables you are having. You know about 5 and you are finding the 6th one from an equation. So either it can be a sample size which is left blank so that you get the sample size from the equation, or the power of the experiment after maybe the power is found out. What was the power of your experiment? So it means how close your experiment is likely to predict the results as a true positive. So that is the thing. And secondly, there is a technique known as Mead's MEAD. Mead is the name of a scientist. Mead's resource equation technique. It is very simple for you to use. I can send you the equation. So it depends upon how many groups or how many treatment groups you are having, based upon which a sample size will be calculated. So you get a statistically for you to get a statistically significant defect. So these two techniques are widely used, and for your specific growth hormone, we have to see which animal model. And again, if you are using an animal model with lower variability, for example, an inbred strain, it won't be as same as what you use, because the standard deviation has come down. So from the equation which I told first, which is the power calculation technique, it automatically gives you with a lesser number of animals if you are using an inbred mice with respect to an outbred animal model in the equation. So for you to do that equation, you should know about the standard deviation that parameter which you are looking out for in your experiment, either through a pilot study or through some others experiments. If you are taking data from some others experiments, you should be sure that you are using the same animal model with the same genetic makeup. or at least you should be confident that these animals will be responding in a similar way in the enzyme system or the growth hormone system you are studying. So that is how you can draw a sample size. And generally, what we see among scientists or in ethical committees doing is that n is equals to 6 as a default number is being taken, which is unscientific. So more explanations I can help you in case you require that. Kindly don't hesitate to write to me.
I will always send my email to you. Please send it to me, please.
Yes,
we have provided.
It is the last slide of the presentation. Once Dr. Manav and the organizers will circulate the PDF, it will be there in the last page.
We have also provided the email of Hari Krishnan sir in the chat box. Those who are interested, they can pick that email ID and mail Hari Krishnan sir directly. Sir, is that email correct?
Yes. Let me see where in the chat box it is running.
At the last, sir.
Okay. So I will type it down.
Okay, sir. Thank you.
You can use either of these things, these addresses. So I'm in home quarantine, room quarantine. I might take some time to respond. Please bear with me.
Okay, sir. Then I think we can conclude. And finally, I'd like to thanks again for accepting our request and presenting a nice presentation, sir, in this web course. And this is an announcement for the participant. We will meet again at 4 o'clock for the next session.
Thank you so much to be with you all.
Okay. Thank you, sir.
Bye.
Okay.